This interview is with Simon Boulton, a member of EMBO and Fellow of the Royal Society and of the Academy of Medical Sciences. He is a senior group leader at the Francis Crick Institute, where his lab specialises in DNA double-stranded break repair mechanisms in mitotic and meiotic cells. Simon is a co-founder of Artios Pharma, a biotech company aiming to selectively kill cancer cells through DNA repair inhibitors. He is also director of RadNet, City of London, a Cancer Research UK initiative aiming to improve radiation treatments across the UK. I hope you enjoy this interview as much as I did. Thank you. Welcome, Simon. It's great to have you on the podcast. So we'll kick off with a question which I always like to ask people of why you chose this career path in particular. So your work focuses on DNA double-stranded break repair mechanisms. Why this avenue over other avenues of cancer research?
I would like to, well, thank you, Martha, for asking me to be involved in this podcast. It's great to be here. I'd like to say that there was some rational design to my career, but I think you would speak to most people and find that actually you just land somewhere and you find something that actually is really interesting. But actually, I think maybe a bit of backstory, which I think reflects the fact that a lot of your career choices end up being really influenced by a number of really defining moments. And mine was actually, I did my first degree in Edinburgh, which is a great place. I love Edinburgh. And I was doing molecular biology, and I was coming towards the end of that, the degree. And I knew that I was really interested in science and experimental science, and we'd had the opportunity, as I think most people do on these courses, to do some lab work, and I'd been in a lab over the summer, and I was thinking about doing a PhD, and it was not clear to me where and what I wanted to do, because I was interested in so many different things, and that's obviously a real challenge, is to focus, right? And actually, I was waiting for the lift doors to close in the building where I was doing my degree, and this arm came through the door as the door clanged, and the door reopened, and in walked one of the professors, Professor Jean Beggs, outstanding scientist who's worked on splicing for many, many years. This is how mRNAs are spliced to produce open reading frames for translation. And anyway, so she and I got talking, and I said, look, I'm really interested in doing PhD, but I have no idea who or what I should do. There's such a breadth of areas. I mean, I wasn't necessarily aligned with doing work in cancer research at the time. I mean, I was very open to doing lots of things. But anyway, Jean said to me, she said, well, there's this young guy who's just returned from doing a postdoc in Berkeley in the US, and he was a PhD student with me. And I think he's going to do remarkably well. Obviously, no guarantees, right? Because it was early on in his career. But she said, look, why don't you reach out to him and ask him if he has availability. So I wrote to this guy. I didn't know anything about him, and he was called Steve Jackson, and he'd probably started his lab maybe 1 or 2 years earlier. So it was really quite early in his independent career. And this was at Cambridge in what is now the Gurdon Institute. It was called the Wellcome CRC Institute at the time. And I reached out to him and said, look, I've been advised by Jean Beggs, your former PhD supervisor, to apply to your lab. I think, what you're doing is really interesting. And he just discovered a protein called COO as the factor that targets a kinase called DNA-dependent protein kinase to double-strand break ends. And to be honest, we really didn't understand at this point what exactly this was doing. We thought it was important because obviously double-strand breaks are very dangerous for cells. They also exist at the ends of our linear chromosomes, right? They end, or they could potentially be seen as double-strand breaks. but we didn't really know what it meant. But I thought this was kind of interesting, right? But as I say, I was interested in lots of things. So I went to interview with Steve, and it was bizarre, you know, because your experience is relative, right? You know, whatever you've experienced in your life so far, that's your point of reference. Well, you know, Edinburgh was great, but the labs I was working in were quite old. When I went to Cambridge, it was like walking into like the 25th century. It was like, oh my gosh, this place is incredible, right? It's so space age, and it was a relatively new institute, and they had all this fancy equipment, and it just blew me away, you know, and I just thought, I've got to be here. This is just so cool. And anyway, I won't go into the the little bumps in the road that led to me being in Steve's lab. But anyway, I started a PhD with Steve, and Steve, we're kind of, it was really interesting because the lab was quite small at the time, but then there were a number of key discoveries came out from Steve's lab in collaboration with a group in Sussex headed by Penny Jago, and they discovered that this DNA-dependent protein kinase, which is targeted to double-strand breaks by Ku, is mutated in a disorder called severe combined immunodeficiency. So children, unfortunately, who are born with this basically lack an immune system, and basically the reason for this is that you need end joining, which is what it turns out what Ku and DNA-PK do. And end joining is basically a gluing mechanism that sticks double-strand break ends back together, independent of sequence. So it can be somewhat error prone. But if you need that actually to repair program double-strand breaks that happen during the development of the immune system. So your immunoglobulin genes, they end up being recombined. So they have, for example, V, D, and J segments. There's multiple V's, D's, J's, and you use a protein called the RAGs. It's a protein complex, and they're essentially specific scissors that induce breaks within these regions, and you repair them in multiple different ways to build a repertoire of V's, D's, and J's. And that is the starting point for the repertoire of antibodies that constitute the immune system. There are additional mutational processes called somatic hypermutation that alter certain parts of antibodies that again expand the repertoire, and that's how we ultimately develop an immune system that can recognize multiple different antigens or foreign bodies and those kind of things. So SCID, basically, these kids are unable to generate the repertoire of antibodies, and that's why they basically lack an immune system and they have to live in a bubble, unfortunately. So anyway, so that's what Steve was working on. So your original question, and I know it was a bit of a convoluted route, but everybody's got a story, is that Steve started to work on Kus, and we weren't clear exactly what they were doing. You know, we had some ideas that they might be involved in repairing double-strand breaks, but that discovery of mutations in SCID was a big part in then the evolution of the lab, and that's how I ended up working on double-strand break repair, because that's what the lab was working on. And it wasn't that I had some divine intervention that this is what I absolutely had to do. It was complete serendipity, and I got very excited about working on it, so that was it.
Yeah, I mean, that's really useful for me right now, because I'm in that position of trying to find maybe a PhD for postgrad and which route to go down and all of that. So yeah, I mean, that's nice to know that not everyone knows what they want to do.
I don't think anybody knows really, right? I mean, I think, once you read around and, I think you guys are so much, I'm showing my age now. We just started to have email when I was doing my PhD. There was, it was, quite a long time ago. But you guys have access to the internet, which can also be daunting because there's so much information, right? And how you sift through that and decide what it is that's interesting, because probably there's so many things. It's focusing in. But I think I would say when you get that gut feeling, I mean, when I walked into Steve's lab and it felt like I was in the 25th century, and it was really inspiring, and I was just desperate to be there. and you know, that was like a gut feeling. It wasn't anything rational. I know that's not very helpful, but sometimes you have to go on gut instinct.
Definitely, definitely. But yeah, so we'll unpack some of the stuff that you touched on in your first answer a bit later.
Sure.
But just for the listeners, just to go straight back to the basics, could you sum up the differences of the two main repair pathways? So homologous recombination versus non-homologous DNA end joining, and yeah, how these are different as mechanisms of repair.
Sure. Yeah. So they're the two most simple. There are other ones that are they're kind of slightly adapted from them, but for simplicity's sake, I'll answer the question that you've asked. So end joining is essentially as described on the tin. If you have a double strand break, which is both strands of the DNA duplex is severed, that can be pretty dangerous, right? And arguably, double strand breaks are one of the most dangerous DNA lesions that can arise in your DNA. The reason being, of course, is that if you don't repair those breaks, you will lose genetic information. So as you probably know, the way chromosomes segregate is through centrosomes attaching to goneticals and segregation of chromosomes to different poles of the cell, and then you get cytokinesis and abscission, right? And so the goal of cell division is to get an exact duplication of your genome, segregate it in two, but the issue, of course, with a double strand break is that the region of the chromosome on the other side of the break that lacks a centromere won't be correctly segregated. So you will get what we call aneuploidy, right? And of course, this is what happens in cancer. And I'm sure we'll go into why double strand break repair is relevant in cancer a bit later during the chat. But what end joining does, as we said, it literally just it basically brings those break ends together and religates them. So the analogy I like to give is just like a glue. If you imagine a Pritt stick, it's basically Pritt sticking the ends together. And actually, the thing about end joining, and it's an important differentiator from recombination, which we'll talk about in a minute, it occurs independent of DNA sequence. So obviously, breaks can happen anywhere in the genome, and that position in the genome has a defined sequence, right? End joining doesn't care. What the Coup proteins do, which we've touched on already, they're a little bit like essentially they're a protein complex with a hole, and they slide onto the ends. So they're there, and DNA-PK and some of the other proteins that we won't go into just for simplicity help to tether those bits together. And then what Coup and various other proteins do is that they ultimately recruit a ligase. A ligase is something that sticks the ends back together. So that's essentially what end joining is. It's relatively simple. It happens throughout the cell cycle, right? And again, this is really important when we talk about recombination. So you obviously have gap phases, G1 and G2. S phase happens after G1, and then you have a gap phase, G2, then mitosis, cells divide, and then you start over again. So end joining can happen at any point during that cell cycle. So how does recombination differ? Well, it's end join. So recombination actually requires an intact sister chromatid or homologous chromosome to repair as a template. So the analogy here is that recombination is a photocopier. So what it does is, if you've got a break in a chromosome, you use the intact sister chromosome as a template for repair. And what happens is that break invades into its identical sister, so the identical chromosome, and then copies the missing information. And that leads to a complete accurate repair. Well, it's a very high fidelity repair of the double strand break. So with this comes a key requirement. You need to have replicated your chromosomes in order to generate a sister. So recombination can only happen once S phase has happened. So as soon as that molecule is replicated and there's now two sisters, And they're actually held together in close juxtaposition by molecules called cohesins, which are laid down by the replication machinery. And the thought is that they actually act as rings that essentially encircle both sisters, so they're held together like this. And so the identical sequence is in close juxtaposition with the other sequence. So it's not like you need to search a long way for the right sequence. They're side by side.
Yeah.
So recombination has a much higher fidelity. It's much more accurate than end joining because it uses identical sequences as a template for repair. It takes quite a lot longer to do as a process. It actually requires the BRCA genes. So again, we're now touching on cancer. So BRCA genes, for those who don't know, there are two major BRCA genes which mutations predispose to breast, ovarian cancer and other types of cancer. And the reason is that if you can't do that accurate recombination, you default to other end joining repair mechanisms, which are more error prone. And we've talked about end joining, but there is a third called MMEJ, or microhomology mediated end joining. And I actually discovered the rudimentary version of this in my PhD. We didn't know what was doing it at the time, but we called it error prone repair that was guided by short microhomologies of sequence. And those actually end up rearranging the genome in a lot of cancers. So if you imagine that if you can't do recombination, the accurate mechanisms, any breaks that arise in your genome end up being repaired by these less sort of more error prone repair pathways. And that leads to translocations, genome rearrangements, and this is really the prelude to acquisition of traits that lead to loss of growth control, ability to invade and metastasize, and sort of all the things that are hallmarks of cancer. So when you look at cancers, you see a distribution of some cancers have very low rates of rearrangements, others really, really high. BRCA deficient cancers are generally have a scrambled genome with lots of rearrangements with hallmarks of these microhomology mediated end joining events. So recombination requires a lot more proteins. It takes longer to do than end joining, but you'd favor it, right? You favor it because you'd like to maintain your genome, but obviously it's better that you maintain your genome to some extent. So end joining pathways exist as the next best situation. It's not perfect, but the long-term ramifications of doing just end joining repair is that you probably have a predisposition to cancer later in life.
I mean, yeah, thank you. You've touched on many questions, which is fab, and how this difference in mechanism can impact lots of different disorders, not only cancer. But yeah, so now we'll go into a little bit more of how you might experiment on and discover things about this. So throughout your time, you've worked on a range of different models, including Saccharomyces cerevisiae, the budding yeast, C. elegans, the nematode worm, as well as mouse models and human cell lines. So what different benefits and challenges do these bring to the table? Why would you choose one model organism over another for different experiments?
Yeah, it's a great question. Well, we can start off by saying there's no perfect model, right? There is no perfect model. They each have their advantages and disadvantages. So when I started my career over 30 years ago working in yeast was a really powerful system, and bacteria were the same, right? You could do genetic manipulation, so you could make knockouts of genes. So you can take them out and then ask what happens when you get rid of a particular gene. And of course, yeast is a single cell organism, bacteria is as well. And so you can study processes, and most of the fundamental processes in life are conserved in bacteria, yeast, and so things like how cells divide, the mechanisms that control cell division, how you replicate your genome, how you segregate chromosomes, and how you repair them are largely conserved. But there are additional complexities when you start moving into higher organisms. And therein lies the issue. Okay, so yeast can be manipulated. You can do it in a relatively short time frame. So your experiments that you do may last a matter of weeks. So that's, it was a really powerful system. But then when you start looking at things in a bit more detail, so for example, the reason I started to work in worms, in nematodes, so the challenges of yeast, of course, because it's single celled, you can't look at really how cell-cell interactions happen. They don't have defined tissues, right? Like the nervous system, the gut, those kind of things, the nervous system, all those things. So you can't study certain processes in yeast. So model systems such as Drosophila, which is the fruit fly, and C. elegans have been used to study complex processes such as how you develop from a single cell into a complex multicellular organism that has, each cell has, well, the cells may comprise organs, they have unique functions. How does that happen? And actually, C. elegans was the first multicellular organism. It has just under 1,000 cells. There's actually it was the first organism to have an entire cell fate map. That is, when you get the fertilized egg, and C. elegans is really interesting, it can do asexual and sexual reproduction. So you can get sperm fertilizing an egg, but the female is actually not a female. She's a hermaphrodite. So she can do both sexual and asexual, and it switches depending on. So this actually is important because it allows you to make clones of things. So if you always do self-fertilization, so using the hermaphrodite zone, then you can maintain clonal populations, but then you can introduce diversity by bringing in males of different genotypes. So this is quite powerful and it's quite unique. Now, the great thing about C. elegans, and the reason I started studying it, was because some of the really important disease pathways or disease genes, and we can talk about the BRCAs. We've already touched on those. There's a pathway, an important DNA repair pathway called Fanconi anemia. So this is involved in repairing damage at replication forks. It's not obviously conserved in yeast. The BRCAs are not obviously conserved in yeast. Well, they're not, right? There are factors that will do some things that are somewhat similar, but the actual proteins are not obviously conserved. So herein lies the issue with the yeast, right? It isn't a good model for studying some of the additional complexities you see in multicellular organisms. You can't study cancer formation in yeast. You can't really study cell death. That's a bit controversial. People have claimed that there is apoptosis, cell death in yeast. Doesn't really make too much sense to me, and I've not followed whether or not where the field is. But one of the things that's really important, actually, C. elegans was instrumental in defining the pathways that lead to programmed cell death, and that might be a little bit difficult to conceptualize, but actually during development there are waves of apoptosis. This is cell death that happen. For example, if you imagine your hand, it may form like this as one structure, and I'm not sure whether this is true, but you need to kill the cells in between in order to form the digits. I'm not quite sure whether that's true, but you get the point that sometimes you need, in order to model organisms, sort of model organs or structures, sometimes you need to kill cells in order for that to happen. And also when cells get damaged irreparably. So this is where, it's very interesting that the mechanisms that actually are really important in cancer that eradicate damaged cells, they're often targets for mutation. And one of the most famous one is p53. It's been termed the guardian of the genome. It's a target for the what we call the DNA damage sensing machinery. So if you get a double strand break, there are proteins that are recruited that basically activate an alarm system. Essentially, they call the police, they call the ambulance, they call the fire service. And what those things are things that will communicate to other cellular processes, such as the cell division machinery, say, look, we've got damage. Don't divide the cell. Stop replicating. We're going to have a pause. We're going to give time for us to sort out the mess. And then if the mess can be repaired, then things restart. If the mess can't be restarted, so what p53 does is it makes the decision between whether or not a cell lives or dies. So it then talks to the cell death machinery to basically eradicate cells that are damaged irreparably. So it turns out p53 is the most mutated gene in cancer, right?
Yeah.
So you get rid of p53, you get rid of that guardian of the genome that basically controls, basically cell fate between damage, repair, or cell death. If you get rid of that, then you have no way of eradicating damaged cells, and then you accumulate mutations, and that's why you get both familial, a disease called Lee-Fraumeni, which is the families basically have inherited mutations in p53 and have a very, very high lifetime risk of developing a whole range of cancers, and then many sporadic tumors that happen within the normal population. They accumulate p53 mutations at a very high frequency, probably upwards of 50%. So yes, C. elegans was actually used as a model to discover the whole cell death machinery, and actually there was a Nobel Prize awarded for that. There was a Nobel Prize awarded for discovering what we called the RNAi machinery. So this is a silencing mechanism that's used in development, but also we exploit it to downregulate genes. And this was actually around the time I was doing my postdoc when I transitioned to work with C. elegans, because RNAi was a really powerful tool. You can basically take any gene of interest, make double-stranded RNA, feed it to the worms, and that particular gene is then knocked out. So you have a way of knocking out genes now, and it's somewhat analogous to what we did in yeast. But in that case, we were actually deleting the gene, physically deleting the gene. Whereas with RNAi, we are basically down-regulating the transcripts of a particular gene, and that gives you a phenotype. So we are able to then basically systematically knock out any gene of interest and then score for a particular phenotype. And this proved really powerful. We call it reverse genetics, where you basically screen for a phenotype systematically by knocking out all of each one of these genes individually. So C. elegans turned out to be really a powerful model, and my lab discovered the BRCA genes in C. elegans, and that was the simplest organism that has BRCAs. There are actually, that's not quite true. I think there are Pseudomonas have them as well, but and trypanosomes, so more simple organisms. But C. elegans was the simplest at the time, and so we ended up working on this for a while, and that was great. And we discovered a lot. We used genetics and reverse genetics, forward genetics, and we tried a bit of proteomics, which is to basically pull out a protein and see what's bound to it, which we use mass spectrometry to try and identify the proteins that are essentially bound to a particular protein of interest. And usually they're the things that proteins interact with, either work with them in a process, or they might antagonize that process, but they could be key regulators, positive or negative, or they might connect that particular machinery to another part of the cellular machinery that's important. You just don't know, but it's a way that we start to interrogate the process of a particular gene product protein. So C. elegans was great, but it doesn't really develop cancer, right? So yes, it has some of these key genome stability processes and factors that are not conserved in yeast. So that was great, and it gave us, a real niche to work in the genome stability field for a while, because it allowed us to do things that others weren't able to do. And the time frame was, a matter of, for yeast are a matter of weeks. C. elegans were weeks to months. But then we started publishing papers on disease-relevant genes like the BRCAs and the Fanconi anemia proteins, and people would say, this is great, but is it true in mammalian cells, right? So therein, we were kind of, our hand was bent somewhat by the review process that we go through in trying to publish our work to basically demonstrate that what we were discovering in C. elegans had utility or value in a more complex organism like humans. So we then started to work in mouse and humans, right? Human cells. Now, of course, they're more closer to us, obviously human cells, but most of the human cells we work with are pretty divergent. They're usually cancer cells that are highly transformed, and they have all sorts of mutations in them. So they're not perfect models. I mean, we're discovering all sorts of stuff about, different cell lines have different mutations that influence processes positively and negatively, and it can be really complicated. So there's no perfect cell. And mice, obviously there's ethical considerations, right? Using mice, people, that is obviously people are against animal research, but I would say that, we completely agree that, any experimentation should be done with very defined regulatory frameworks, and where possible, we try to minimize animal usage. But there are certain things we cannot do in a tissue culture dish. And when we're studying cancer and cancer predisposition, we have to do it in an animal, right? And I think people understand that if we're going to develop therapeutics that are beneficial to human treatment of cancer, we have to, and the legislation is such that we have to do these things in animal models. So mouse is one of the main ones that we use. And of course, so we've discovered the ethical issues, but also the timing issues, right? Cancers in mice can take anywhere between, several months up to over a year to develop. So the length of the experimentation is considerably longer. They're much more expensive. They're much more complicated. They obviously require husbandry and being looked after very carefully. And, as you would expect and would hope, that they're treated with complete respect. But they're being used to study key processes that might have relevance to human disease. So I think you can probably gather already that each model system has its own strengths and weaknesses. In the case of mouse, we can study really complex cell-cell interactions, organ interactions, how the immune system relates to different processes, how that influences cancer predisposition, the immune system, and how it recognizes foreign bodies, really important component of cancer. You can't study that very easily in a cell culture system. We're starting to develop co-culture models where you can do this, but it's not. It's a reductionist system. It's not the entire picture. So you really do need to study it in more complex systems. And human cells, well, we can now re-engineer. We can make what we call induced pluripotent cells. So was discovered a while ago, factors which we call the Yamanaka factors can be used to reprogram cells back to their sort of primordial, I said it's not really primordial, but their most naive state. That's the word I was looking for, naive. So that initial perfect cell which forms when the egg and the sperm come together, that which is fully pluripotent. What that means is it can divide and produce any cell that will ultimately give rise to you. So for that to happen, that cell needs to make a choice. So it's about self-fate decisions. Cells must differentiate and become gut cells, nerve cells, skin cells, etc. Right? So they have differentiated. So what you can now do using the Yamanaka factors, you can reprogram cells back to that naive state, which has essentially a fully pluripotent capability, and then you can then differentiate those to different cell types, right? You can make different types of neurons, different types of gut cells, different types of, you can make cardiomyocytes that are actually beating in culture. It's really cool. So, but this is technology that's probably only really happened in the last sort of 10, 15 years. And it's challenging to work with. And then we have 3D models now, which we call organoids, which are three-dimensional structures that also have certain different cell types within them. But again, they're not perfect, right? They're a reductionist system to study processes. So it's all about timing and understanding what it is, so timing being how long it takes to do certain things, right? So engineering cells is now possible with CRISPR technology. We can introduce very specific point mutations. We can delete whole genes. We can introduce tags into proteins. We can rearrange the genome in very specific ways. But this was only really possible in the last 5 to 10 years. So things have changed. But why we've worked on different organisms is because our view has always been to try and exploit the unique capabilities of that organism with the knowledge that they have limitations. So we always try and study in multiple different systems, whether it's cell culture, in C. elegans, in mouse, exploiting their respective strengths, and that's why we do it.
Yeah, absolutely. And using the different combinations of different model organisms and knowing the limitations of each of them, and then using the knowledge that you gain from that in combine, that's really important. Yeah, so in that answer you touched on P53. We'll cover that maybe a little bit later. But just to jump slightly back to your work on budding yeast, you looked into telomeres and the roles that these have and the regulation. So what did you discover as the link between non-homologous end joining pathway, as we've discussed, and the maintenance of telomeric length and telomeric transcriptional silencing?
Yeah, this is a long time ago. I'll have to see if I can remember. No, I do remember. But yeah, this was in my PhD, nearly 30 years, actually over 30 years ago. Yeah, so this has been Steve Jackson's lab. We discovered a number of the end joining proteins in yeast, and we started to study them. And I made I suppose there were two key things. Well, there were a couple of things that I think were important from what I discovered in my PhD. The first was we developed a very simple system to measure double-strand breaks. We used a plasmid DNA. These are circular DNA molecules. And what we were able to do is just introduce breaks with restriction enzymes, so special scissors that cut specific sequences. And we could then introduce those into cells and look at how efficient they were at repairing, because they carried a selectable marker on the plasmid, so you could basically plate the yeast on a media lacking a particular nutrient, and that would be provided by the plasmid as long as it was repaired. So this was a really simple assay, but it turned out to be really powerful because it allowed us to show that plasmid rejoining is absolutely dependent on end joining, non-homologous end joining. And the other thing that you can do is, of course, you can take the plasmid back out when it's been repaired, and you can sequence it to see whether or not there are mutations or any changes in the repair. And so the first thing we showed was that compared to normal yeast, which we call wild type, that can repair these plasmids really efficiently. If you're end joining deficient, so deficient for Q, ligase 4, several other proteins as well, then the ability to repair those plasmids plummets, down to 1 or 2% of what you see in the wild type situation. And what that was really interesting, and when we recovered the plasmids, what was interesting is that some did get repaired. That 1 or 2% got repaired, and when we sequenced the breakpoint where the enzyme had cut, we discovered deletions and insertions. And when we looked at the breakpoint, we discovered these microhomologies. That is short identical sequences. It could be GAT, for example, or GGG or whatever. They're just small sequences that could pair, and then you would get deletion of any intervening sequence. And so this was what we call we called it a backup error-prone pathway.
Right.
And so this kind of hinted at the existence of a third pathway of double-strand break repair. The other we talked about is recombination, and if you knock out recombination, you also see this backup pathway. So we discovered this in yeast, and we called it error-prone double-strand break repair that happens when you don't have end joining or recombination. And it took another 20 years for us to discover that this pathway is actually driven by an enzyme called Pol theta. And Pol theta is a critical, becomes critically important in cancer cells, particularly those that are deficient for recombination. And actually, my company that I started, Artios Pharma, about 8 years ago, we just celebrated our eighth birthday, has developed the first Pol theta inhibitors for treating cancer. So they are currently in phase 2 clinical trials. And we'll see whether or not they do. I can't really mention exactly confidentially, but we're very excited that they may provide an opportunity to do lots of things to treat certain types of cancer. But anyway, so that's what we discovered in yeast a long time ago. We've discovered this error-prone pathway. But at the same time, we also found, because of course, chromosome ends. So most organisms have linear chromosomes. We have linear chromosomes. Yeast have linear chromosomes. Bacteria tend to have circular chromosomes, so they don't have an end. So ends create a problem, right? Because they could be seen as a double-strand break. And so cells have evolved specialized structures called telomeres. And these are they differ somewhat in different organisms, but simplistically, what generally happens is that you have a highly repetitive sequence. In humans, it's TTA-GGG that's repeated multiple times, and they comprise the telomere sequence. And in us, they can range from like 10 to 20 kilobases in length. So you have that many repeats at the ends of every chromosome. And what they do is they serve as binding sites for very specific proteins that help to maintain the chromosome end. So there is a complex called sheltrin that was largely discovered by an amazing woman, one of my heroes in science. Hope she doesn't hear this. She probably knows it anyway. Tizia de Lange. She's at Rockefeller University in New York, and she's a real pioneer in this space, discovered many of the proteins that bind to human telomeric sequences. So sheltrin is a 6 subunit complex, and basically it helps a number of things. It helps the replication machinery to replicate through the telomere, because repetitive sequences have a tendency to form secondary structures that can impede replication. There's also so that's one thing. The other issue is that the replication machinery can't replicate to the very end of a linear molecule. And your listeners may know that replication is semi-conservative, and it has a leading and a lagging strand. And it turns out that because of the lagging strand synthesis, you can never replicate to the very end. And so as a consequence, telomeres shorten with each cell division. And this actually defines how long your cells can divide for. So it defines your replicative lifespan. So if you are born with really short telomeres, you're going to age quicker, right? But if you have really long telomeres, that doesn't mean that you'll live forever. It actually creates a bit of a problem as well. So there is a Goldilocks length that is optimal. If you have really long telomeres, you tend to be cancer prone. And the reason is that when our cells differentiate, so our stem cells, which I'm sure you guys will know about, these are somewhat pluripotent, so they have the ability to adopt different, produce cells with different fates, whatever. We talked about that earlier. So stem cells need to be maintained long term. So they have an enzyme called telomerase, which is a reverse transcriptase that can add telomeric repeats, so you can extend the telomere out to a certain length. So this addresses this end replication problem, which is the problem that the replosome, the normal replication machinery, can't replicate to the very end of the chromosome. So telomerase can do that in our stem cells. But when our cells differentiate to become, more defined cell types, such as neurons or skin cells or kidney cells or heart cells, whatever, they downregulate expression of telomerase. And the reason is because this end replication problem actually serves as a barrier to cancer. So you can imagine that cancer cells can want to divide indefinitely, but it's not that they think that they want to divide. They just, in order to grow indefinitely, to form a large mass, they need to overcome certain problems. And one of those problems is the end replication problem, because that would basically limit the cancers to dividing, you know, let's say 50 or 60 times, let's say. So the way cancers solve that is to re-express telomerase. They select for clones within the cancer that will now express telomerase again. And that therefore then can solve the problem because it can add repeats indefinitely. So that's one of the things that sheltering does, is it regulates telomerase, helps to recruit it, defines the length of the telomere, and that's an area we still don't fully understand, is what defines the length of the telomere that we each have. But the other thing is that, as we've discussed, and why we were interested in it, is that the telomere ends can look like a double-strand break. And actually, it was Tisha de Lange who discovered that one of the sheltering proteins, these are the proteins that bind to telomere repeats, a protein called TRF2, if you take it away, so if you delete it, you see this incredible phenotype. It's just remarkable. So if you look, all of your chromosomes end up fusing together by their telomeres, by end joining, by Q-dependent end joining, to form massive long spaghettis of chromosomes, all linked end to end, right? So this is called the end protection problem, right? So essentially what sheltering does is it prevents the end of the chromosome from being seen as a double strand break. So, and you can imagine, if you fuse all your chromosomes, you're going to die, because when you try and segregate them, you're just going to pull the chromosomes apart. So it becomes a real mess. So it's incompatible with cell viability. So this is a really important problem. So what we discovered in yeast, and this was way before any of what I've described, right? We didn't know a lot about telomeres. I think we understood about telomerase. We were starting to understand various other processes. But what we found is that, you know, Q being an end joining factor, it's actually recruited to telomeres. It's present there. And this was a bit of a paradox, because, you know, telomeres, if they're bound by Q, why don't they fuse? And we didn't really know what that meant at the time. But what we did discover is that the telomeres rapidly shorten to a new set point. So they don't progressively shorten like you would happen if you knock out telomerase, which just gradually shortens over time until you hit a crisis where the telomere is no longer functional. But with the Q knockouts, and this was also true for other components that we discovered, they shorten by a certain point, and then they maintain at that shorter length. And you know, this was a descriptive, but now we saw that this was what was happening, and at the time we had no idea why this was happening. And other people in the telomere field have gone on to really define what Q is doing there. Actually binds to the RNA component of telomerase and can do various things there. So we observed this, and I think what was perplexing is why you've got DNA repair proteins at telomeres, right? And yet they don't fuse, or they don't cause problems. So that was a bit of a mystery, but this is true. And it turns out that quite a few repair proteins end up moonlighting. They do additional functions at telomeres. Whether it's helping to process the end in a particular way so that you can extend it by telomerase, or I've not quite told you the whole story, but the ends of chromosomes don't have a flush end. They actually process the end back to have a 3' single strand overhang. So there are lots of additional complexities of telomeres that we've not gone into. But that's what we discovered in yeast. We also discovered that Qs are important for silencing. So this is basically shutting down transcription within the subtelomeric region. This had been defined for what's called the SIR proteins, and we found that the Q and the SIR proteins were involved both in end joining and at the telomere. We discovered another complex called MRN. We didn't discover MRN, other people did, but we found that it was important for end joining and was also important at telomeres. So this was really interesting. So I think the key points from my PhD were this plasmid rejoining assay, the discovery of this error-prone pathway that ended up turning out to be really important in cancer, and also this unexpected discovery that repair proteins are at telomeres and do stuff at telomeres that quite frankly we didn't understand at the time, and others over the years have discovered what they've done.
Yeah, so you've really revolutionized the way that we view telomeres and how the repair mechanisms interact with this. And yeah, so just really quickly before we move on to your most recent work, just for time purposes, could you quickly sum up, and we'll go more into the double-strand repair mechanisms more specifically in the proteins that are involved there. So there's two of these proteins, p53 binding protein 1 and BRCA1, which you've discussed already. These have a mutual antagonism that help sort of the cell decide almost which repair pathway to go down. So could you just really quickly sum up that?
Yeah, absolutely. So yeah, 53BP1 and BRCA1, they are critical components, that factors that define whether you do end joining or recombination. 53BP1 is pro end joining, whereas BRCA1 is pro recombination. And it all comes down to one key thing, and that is how you process the ends of your chromosomes, right? End joining likes a blunt end, right? Yeah. So end joining likes to basically have a substrate where you have a flush double strand break end that they can then religate. There can be some little changes that happen, some processing of the ends, but it likes a blunt end. In contrast, recombination, you need, in order to make a substrate that can invade into the identical sister chromatid, you actually have to process that end. Similar to the telomere, you have to process it back to produce a 3' single strand overhang. So you actually process the end back. It's called resection, and there are lots of proteins that have been discovered that that basically help facilitate that processing back of the end. And it's that single stranded DNA that's then bound by a key protein called Rad51. And my lab has worked on this for many years. My neighbor, Steve West, is one of the pioneers of this field, has worked on Rad51 for many years. It's and this is the substrate for recombination. It forms a helical protein filament on that single-stranded DNA, and then has the ability to identify the identical sequence in the sister chromatid. It can then invade and pair the broken molecule with the complementary sequence in the sister chromatid, and that sets up a replication fork that can then copy the information. So end joining likes blunt, recombination requires processing, and 53BP1 and BRCA1 regulate that choice, and it's controlled during the cell cycle. It's impacted by the key kinases that control cell cycle transitions. They're called cyclin-dependent kinases, and phosphorylation events. These are post-translational modifications of proteins that are regulatory in nature, control whether or not you do recombination or end joining. So what I've told you already is that you can only do recombination in S phase. Once you've replicated, you can do SG2. So in that scenario, so pre-S phase in G1, 53BP1 dominates, right? It actually blocks resection in a number of different ways, favoring end joining. But then when you transition into S phase and you now start to get increase in certain CDK activity, that switches the repair pathway to pro recombination, where you can now resect the break, and that is now through BRCA1. So there's actually a paper just came out in two papers came out in Nature describing the sort of the biochemical mechanism by which BRCA1 controls resection, which is really interesting. So they're kind of mutually antagonistic, but they play key roles in this pathway choice, is what we call it.
Yeah, amazing. So just really quickly, in order to test the ability of cells to carry out these different double-stranded repair mechanisms, your group are using reporter assays via detection of nanoluciferase luminescence. Can you go into quickly how this works and what results you're seeing in response to different stimuli?
Yeah, so I mean, we've talked about the different repair pathways. We talked about end joining. We've talked about non-homologous end joining. We've talked about pol theta or microhomology mediated end joining. There's recombination. There's also synthesis dependent strand annealing. There's SSA. There's other pathways. And what people have developed, and we're not the first, by the way, there are a lot of other people in the field have used multiple different tools, whether they're restriction enzymes or existing enzymes like HO in yeast or a CRISPR, Cas9, for example, to induce breaks in the genome and then look at how they're repaired, right? So what these, and so there are tools that we've been using for decades that allow us to distinguish between different repair pathways. These nanoluciferase-based reporters, which were actually developed to Artios Pharma, the company I helped to found back in 2016. So they developed these nanoluciferase reporters that allow you to very accurately distinguish between the different types of double-strand break repair pathway. And then what there then can be used to then interrogate effects of specific drugs. So, you know, if you make a pol theta inhibitor, you would expect that if it was specific and potent, it should very effectively inhibit microhomology mediated end joining, but should have no overt effect on canonical end joining or recombination or SSA. And so these tools are being used really to test the validity and efficacy of these drugs that the company have been developing.
Amazing. And so just to finish off, what are some unanswered questions that still puzzle the field and you think might hold something exciting for the future?
I mean, there are so many questions. When I started over 30 years ago, you know, we thought, oh, this is how a particular process works. But there is so much complexity in biology. I mean, there are still new factors that are implicated in end joining. We're still discovering how recombination works. It takes a long time. Science is, you know, it's not that we're slow, it's just very complicated. So there are many questions, you know, how different repair factors are controlled in different organs. You know, how can we exploit them therapeutically? There are many questions we still don't understand. For example, you know, how precisely how chromosomes protect themselves. We know that sheltering's important. We know that you need to form a loop. We're not quite sure how that happens. You know, there's a huge level of regulation that happens that it almost certainly differs depending on whether you're differentiated or you're pluripotent. I mean, honestly, I could speak for days on the questions that we don't understand. And it can be quite daunting, right? Because, you know, what do you choose to study? And it comes back to the original question, right? I just think you've got to find something that you're passionate about and just give it a go and see where you get to. But you know, I mean, you know, we have a lot more tools. Technology's really driven science forward. Sequencing is now commonplace. We can sequence genomes very rapidly. We can engineer cells down to single nucleotide changes. We can develop three-dimensional models. We can do really complex omics, looking at the entire proteome, the entire transcriptome, you know, all of the post-translational modifications. We're still discovering new ones all the time. I mean, biology's, I mean, it's remarkable, right? How complex it is. Um, but we're aided now by, obviously now machine learning is becoming a big component, artificial intelligence. A real remarkable revolution has been the ability to predict with pretty high accuracy the three-dimensional structures of a protein from its primary amino acid sequence. And that's been driven by DeepMind, which came from Google, and they've released what's called AlphaFold. This is an algorithm that allows you to predict secondary structures. They've released a number of iterations. There's AlphaFold. There's a recent one actually allows you to dock in small molecules into proteins. So this, I suspect, will help drive drug discovery. You can look at protein-protein interactions. You can look at protein RNA, DNA interactions. This was a pipe dream even a few years ago. I'm sure DeepMind were working on it already, but to most people who aren't structural biologists, this was the holy grail essentially, and it will change biology significantly, as has the likes of gene editing and the various tools we have. So science is advancing rapidly. Really exciting time to be in science, as it always has been, right? Again, we talked about relative experiences, right? When I started my lab, oh my gosh, this is amazing, we can do this, right? But then, if I'd been able to look to the future and seen what we can do now, it's changed beyond all recognition. So it's super cool to be a scientist. It's not a job, right? It's a hobby. So I'll probably leave it there.
No, absolutely. Yeah, it's very exciting time to be joining science as a young researcher, and myself. Yeah, you touched on a lot of the things, which is very exciting for the future. So yeah, okay. Thank you so much for joining us today.